Alright, Mr. P here. In the last video, I kind of talked through a lot of those objectives that we were going to cover, and then decided to cut it off at 10 minutes. I just talked about the cell theory. I didn't want the video to get too long, and so in this video, we're going to kind of try to finish out the rest of those objectives, starting with exceptions to the cell theory, get into a little bit about characteristics of life, talk specific application within two organisms that are unicellular, to those characteristics of life, and then kind of finish off with that in-depth discussion of surface area to volume ratio. So, exceptions to the cell theory. We already talked about what the cell theory is in the previous video. If you haven't seen it yet, go back and watch. But cell theory is a theory that states kind of three statements, right? It has three statements that are included in cell theory. Cell theory states all living things are made of cells. Cells are the basic unit of life, and all cells come from pre-existing cells. But there are always exceptions. It seems like there's always exceptions to the rules. There's always gray area, and IB would like you to know that there are specific examples that kind of go against what is traditionally cell theory, or what traditional cells typically exhibit. And so the three exceptions to this cell theory that I want to highlight are muscle cells, aseptate hyphae, which is basically a fungus, and the giant algae and we're going to get into more detail here, but just right off the bat, muscle cells, a fungus, algae are exceptions to the cell theory, and these exceptions have really come about as the result of technological advancements and advancements to the protocols that we've used to identify cells and studied cells. So let's jump into exactly what all of these are. Striated muscle, your muscle fibers. Okay, here's obviously a real image. Here is an artistic rendition of what that muscle fiber looks like. Why are striated muscle fibers an exception to the cell theory? It's all about having multi-nucleus or multinucleated. These cells are different from other cells, most other cells, in that most eukaryotic cells have one nucleus per cell. These particular cells are very long and are actually multinucleated, and sometimes the nucleus or the nuclei can be as numerous as several hundred within one cell. So, what is striated muscle fiber? It's a type of tissue that we use in our bodies to move. Okay, muscle fibers are actually tissues that are composed of these muscle cells that are stretched between two bones that work a lever most of the time, right? You can bend your arm at your elbow because your bicep and tricep relax and contract kind of in concert with each other to move your arm in a very controllable way. The building blocks of this particular tissue are muscle fibers. So these muscle fibers are cells or are structures that are very similar to cells, and they're surrounded by a membrane just like other cells are. They are formed by pre-existing cells like other cells are. However, because they have multi-nuclei or multiple nuclei, they can be much larger than most other animal cells. And again, instead of having one nucleus, they have several hundred. So you can look, this is one particular cell or a section, okay, it's just one section of a particular cell, and you can see that there is a nucleus here, 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 and so that's just one small bit or one section of the muscle cell, but you can see that within every single one of these cells there are a variety of nuclei in each of the frames for each of those cells. And so what does that mean for the particular muscle cell? It just means that there are multiple nuclei that are perfectly capable of running the cell in a very efficient way. Again, it has several things that are in common. Okay, it shares several similarities with other cells in that they are produced from pre-existing muscle fibers, right? Just like other cells. They are surrounded by a membrane, just like other cells. They have organelles and a nucleus, just like other cells, but they are much larger and they have multi-nucleus, okay, or there are multiple nuclei, which kind of goes against what is traditional cellular for our understanding, especially related to the cell theory. So those are an exception. Another exception to cell theory is this fungal hyphae. Now, these hyphae are kind of long multinucleated structures just like a muscle fiber, but instead of being just surrounded with a nucleus, they are actually surrounded with a cell wall, which makes them very rigid, unlike muscle fibers. Okay, if our muscle fibers were surrounded by a cell wall, especially chitin, just like these hyphae, we would not be as movable. Okay, we would be a lot more rigid, much like a fungus. But unlike muscle fibers, also they have kind of these projections that are kind of partitioned in some instances or are non-partitioned in some instances. These partitions help to kind of structure the hyphae from inside. But like muscle fibers, which is again why they are considered an exception, is that they are multinucleated like the muscle fiber. Okay, so far, all cells, or most cells, 99.9% of cells, would have a single nucleus, right? If they're a eukaryotic cell, single nucleus, a membrane, organelles, and they would be produced by existing cells, right? They would just be your traditional, typical eukaryotic cell. So far, we have two exceptions, the muscle fiber and fungal hyphae. Both are exceptions, most notably because they are multinucleated. Okay, multinucleated is very different from traditional cells because traditional eukaryotic cells only have one nucleus. So the last one to kind of round out this series of examples that kind of go against what is traditionally called for by the cell theory is this giant algae. Now, these giant algae are very, very large organisms that are phototrophic, okay, or not, they feed themselves, right? They go through photosynthesis, and so they are like other aquatic plants. They store their genes inside a nucleus, just like any other cell, but they are much simpler in organization and structure than plants because they are essentially unicellular. These fungal, or these algae organisms are single cellular, but they are actually the same size as some other aquatic plants that are multicellular. So again, they're an exception to the cell theory because we've only kind of again brushed the surface of what cellular biology is. We're going to dive into kind of ultrastructure of cells. We're going to get an in-depth look into what these ultrastructures do, okay, structure and function of these ultrastructures, but these are very different. This is an aquatic plant that you can see with your naked eye, much like an aquatic plant that is made of one microscopic cell, and there are tons of these in the oceans, and they form the basic item of many marine food chains, but they grow much larger again than other single-celled organisms, right? Single-celled organisms are traditionally tiny. They're a single cell. Protists are all tiny. Single-celled bacteria, all tiny. All of the individual cells within your body that make up a multicellular organism are tiny. Cells are very small. But you have these giant algae, okay? Giant algae, single-celled eukaryotes or single-celled organisms that are much, much, much larger than other cells. Again, exception. And so that kind of rounds out those exceptions. 3 exceptions, 2 are multinucleated, 1 is very large but unicellular, all 3 very different than other traditional cells. So, we've talked a lot about cells, we've talked about cell theory, we've talked about some examples of organisms that kind of go against cell theory, there are exceptions to the cell theory, but all cells, whether large or small, whether unicellular or multicellular, all have a particular set of functions that are specific to what we have characterized as being alive. In order for an organism to be considered alive, including a single-celled organism to be considered alive or be considered living, they have to exhibit all 7 of these particular factors of life. And they are metabolism, reproduction, homeostasis, excretion, growth, response, and nutrition. Here I have kind of some definitions of what they are. We're going to go through these a little bit, and then I'm going to get into some application of these particular facets of life so that you can kind of see how an organism might actually go through these or how they they actually utilize these factors. But metabolism, as you've seen in previous videos, is the sum total of all enzymatic catalyzed reactions, right? Metabolism is a combination of all anabolic and catabolic reactions in a cell or organism. Metabolism is required in order for an organism to be considered living. Reproduction. An organism has to reproduce and produce viable offspring in order to be considered living. That just makes sense. If you cannot reproduce, you are not, by definition, living. A perfect example, a virus, a lot of people think are living. Okay, it's just like a bacteria cell, right? But it's not, because virus particles, essentially what they are, cannot reproduce on their own. They require infecting a host cell. That host cell has to produce more DNA molecules for it. It has to produce more protein coats for it, and then it has to basically do its reproduction for the virus. Organisms have to be able to reproduce on their own. Homeostasis is the maintenance of a constant internal condition. One of the ways that humans, for instance, maintain homeostasis is that when they experience a situation in which is cold or the core body temp drops, then the body induces shivering, right? Shivering increases friction, it increases the amount of heat at the surface, and can actually warm an organism or raise the internal temp. Like, that maintains a constant internal temp. Excretion, you have to be able to get wastes out. Metabolism, basically the sum total of all enzymatic reactions, the catabolism and anabolism, produce waste products. Those waste products have to be excreted, or they will actually produce toxins and increase in concentration within the cell, and can cause a toxicity that will kill the cell. The cell has to be able to get those toxins out. Growth, living things grow. Very simple. Growth is basically the change in size or shape. It is not maturing, it is growing. It is literally getting bigger. Response, you have to be able to respond to your environment, right? The environment is always changing. You have to stay kind of in concert with a changing environment. If you cannot respond to your environment, for instance, migrate south when it gets cold, or migrate north when it gets hot, right? That is an item that is, or an action that is in response to a particular environment. You have to be able to respond to a changing environment in order to stay relevant. That is a factor of life. Nutrition, you have to be able to feed, and that could either be feeding yourselves through photosynthesis, or can be consuming others through feeding heterotrophically, but you have to be able to feed yourselves. So, to be considered living, again, IB wants you to know every single one of these: metabolism, reproduction, homeostasis, excretion, growth, response, nutrition, all have to be demonstrated at some level by an organism, for that organism, by definition, to be characterized as alive. Okay, so let's look at how other life forms that are very different to us, because it's very easy to look through that list and say, yeah, we do that one, check, 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 all 7. And it makes sense. We're alive. We know we're alive. We know how we handle our environment. We know how we respond to our environment. We know that we eat and we reproduce and we have a metabolism. Okay, all of that is fine and well. But do unicellular organisms go through those as well? And the answer is yes. If we zoom in on this Paramecium and we talk about this being a unicellular organism and the fact that it is alive, but does it exhibit all the factors that we've identified as being necessary to be classified as alive? And the answer is yes. So how does it use excretion? Okay, this is a single-celled organism. It has a metabolism. It produces waste products, and the waste products are actually expelled by the plasma membrane. All of these waste products are going to just naturally diffuse out, and good reactants, things that our metabolism needs, are going to be either consumed through the oral groove, which we'll talk about, or will diffuse in through the membrane as well. But the cell membrane for this particular organism not only serves as a boundary, but also serves to regulate entry and exit of materials. One of the things that is going to exit is waste products. Response: this single organism can respond to its environment in very specific ways. Notice that it has all of these cilia around the outside of the cell membrane, and those cilia will actually detect movements, or can actually move the paramecium in response to environmental changes. These parameciums can actually move to or away from light. It's called phototaxis. It can move towards and away from chemicals. That's chemotaxis. But it can respond to its environment in very specific ways. Homeostasis. There is a contractile vacuole, which you can see right here. That contractile vacuole will fill up and release water based on the environment that the paramecium is living in. So if this particular environment becomes slightly hypotonic, then water will likely come in, and that's basically just an osmotic, or in osmotic terms, a way to equalize or reach equilibrium within its environment so that there isn't an osmotic potential created. But that is just a way for this particular organism to maintain homeostasis, right? A constant internal condition, a constant internal water concentration, so to speak, maintain hydration within its environment. Again, homeostasis. Metabolism. Most metabolic activities occur in the cell cytoplasm. This is a single cell. It has all these organelles, which I know kind of are all running together, but there is cytoplasm in and around the organelles within the membrane. And most of the anabolic and catabolic activities for that particular cell that constitute this total or sum total of all enzymatic reactions make up the metabolism. And again, most of that happens in the cytoplasm. Growth. This organism physically gets bigger. It starts out very small, and it can grow to be relatively large for eukaryotes, right? Single-celled eukaryotes. In fact, Paramecium sometimes can be seen with the naked eye. Definitely can be seen readily with a light microscope at pretty low magnifications. Nutrition. There are food vacuoles inside of this particular organism where food, water, and waste is stored. And they store them within these vacuoles because they actually have to break them down for the organic molecules that are contained within those food molecules. Reproduction, these cells reproduce via asexual and basically just kind of reproduce via binary fission. Again, one becomes two, but they still reproduce. So you can see that all of those functions of life are exhibited within even a eukaryote or a single-celled eukaryote, much like people, which are multicellular organisms. So what about a single-celled plant cell? Much the same way. I'm going to go through this faster because a lot of them are the same way, right? Excretion, just like the paramecium, is going to diffuse through the cell membrane. A lot of the requirements for photosynthesis, like CO2 is going to diffuse in. Oxygen is a waste product, so it's going to diffuse out. That's what photosynthesis is. The diffusion of those products go through the cell membrane. That is a form of excretion. Metabolism, again, happens in the cytoplasm. Metabolism is that totality or sum total of enzymatic reactions made up of catabolism and anabolism. Again, cytoplasm. Nutrition, they are a photosynthetic unicellular organism, so they produce their own food. That food is glucose, just like any other plant. Produces that via photosynthesis. We're going to talk in depth in a little while, okay, in later videos about photosynthesis, so stay tuned. Growth, these cells get bigger, just like the other unicellular organisms we talked about, like Paramecium. Okay, when they get too big, they will split from 1 into 2. And again, that's just to maximize surface area, which is going to be later on in this video as well. Homeostasis. Just like the Paramecium, they have a contractile vacuole. You can see it here. Basically, where they store water, it can fill with water. It can regulate the concentration of water within the cell. And really important when maintaining homeostasis for a changing environment, because environments aren't stable, right? You have to be able to fluctuate. You have to be able to kind of differentiate your concentrations to help kind of balance out what the environment is doing. And again, response. Chlorella have cilia, which are kind of not as visible as in the Paramecium. They're a little blurry in this particular image. But they can move around their environment, just like the Paramecium, to and from light. Again, I called it phototaxis in the last slide. I'll call it phototaxis here. But when you move in response to an environmental stimulus, like light, that is called phototaxis. Okay? And reproduction. One cell can, via mitosis, split into two. Again, reproduction. All 7 of the characteristics or the factors of life can be demonstrated by unicellular organisms as well as multicellular organisms, but it doesn't even matter if it's a eukaryote that feeds heterotrophically or it's a eukaryote that feeds itself via photosynthesis like this Chlorella does. All living things exhibit all 7 characteristics of life. It's really important. Okay, so do all life forms show the functions of life? The answer is you betcha, right? You betcha. So, to kind of bring all this home, right? I know I'm talking fast. I'm trying to keep the video short. You know, if you need to pause, if you need to rewind, if you need to slow it down, please do. Surface area to volume ratio is vital. It is absolutely important when we talk about the limitation of cell size. I have 3 specific size cubes, and it's really important to kind of understand how to find surface area and how to find volume so that we can kind of easily demonstrate what surface area to volume ratio is and how it's calculated. If we have a cube that is 1 by 1 by 1, right? Centimeter, millimeter, foot, doesn't matter. 1 unit by 1 unit by 1 unit. The surface area is 6, right? Because 1 times 1 times 1 makes, or 1 times 1 makes 1, but you add all the sides, you have 6 sides, times 1 gives you 6, right? Volume is when we do the 1 times 1 times 1, which means 1. So the surface area to volume ratio is a 6 to 1 ratio. If we look at a cube that is 2 by 2 by 2, the surface area of this particular cube is 2 times 2, which is 4, times 6 is 24, and the volume, 2 times 2 times 2, is 2 times 2 is 4 times 2, 8. Right? Volume 8. Now, if we simplify that, 24 goes into 8 3 times. You can see that simplifying that goes to a 3 to 1 ratio. So what did we do? By doubling the size of our cube, we actually decreased our surface area to volume ratio by half. Okay? So we doubled in size, but our ratio got smaller. And then if you were to compare this, 3 times 3 times 3, it would be surface area. 3 times 3 is 9, times 6 is 54. And so if we go 54 over the volume, which is 3 times 3 times 3, 27, and we simplify that, that is a 2 to 1 ratio. So what did you notice? We go from the smallest cube being 6 over 1, 6 to 1 ratio, doubling it, dropping it to a 3 to 1 ratio, getting bigger, goes down to a 2 to 1 ratio. So what can you tell me? As basically cell size goes up, the surface area to volume ratio goes down. They are inversely related. As cell size goes up, surface area to volume ratio decreases. And it's very easy to see using these ratios as examples, right? So why is that important? Okay, what are the benefits of having large surface area to volume ratio? Again, having large surface area and keeping volume low. If surface area is our numerator and volume is our denominator, right, we want to keep that bottom number low so that we're not dividing the high or the top number by a lot, right? That's how we keep our surface area to volume ratio large. What does it mean? Smaller cells have physically shorter diffusion paths, right? Diffusion paths are actually shorter. If we put a ruler up here and we measured this distance, that is half the distance of this one. And this is smaller, shorter than this distance. In fact, this smallest one is a third the distance of this one. So this distance is half this distance, and this distance is a third this distance. Either way, of these 3 size cells, this particular cell is the most efficient because the particles that diffuse from the membrane to the very most inside portion of that cell only have to travel that far as opposed to these distances, right? Molecules don't have to travel as far, which increases efficiency. Concentration gradients are easier to generate, which increases efficiency as well because, and here's just an example, it takes less solute to make a 5% solution in a 100 ml beaker than a 5-gallon bucket, right? If I have a little 100 ml beaker, okay, this tiny little beaker, and I put a solution in here, it's going to take less solute particles to make a 5% solution than it does to fill a big bucket of solution and all these particles to make it the same 5% solution. Okay? There's physically more dots, more solute here than there is here. Concentration's the same, but it takes more solute, which means it's not as efficient. This cell can be more efficient with a less less solute, right? It's just like smaller cars are more fuel efficient than larger cars because they take less gas to operate, right? Increased efficiency. You can see here, not only the diffusion paths are shorter, but surface area increases fast. Okay, it goes 1 times to 4 times to 9 times, but volume increases faster. Volume increases from 1 times to 8 times to 27 times. That is a much bigger increase than this. They both increase, but this is a faster increase. So if we are increasing our top number slower than we're increasing our denominator or our bottom number, you can see that very quickly that bottom number can overtake our top number, and that leads to a shorter or a smaller ratio. So, how do organisms maximize their surface area to volume ratio? This is kind of where we're going to end the video. We're going to talk about these specific examples. All of these examples have one thing in common. It's structure dictates function, but also these are structures that are employed by an organism to maximize surface area to volume ratio. First one being cell division. Cell division. Why does cell division increase surface area to volume ratio? And it has to do with decreasing the volume. When we have a large cell, a single cell, and that single cell can split in the middle and produce 2 smaller cells that take up the same physical space, but both of these can diffuse at the same time, and both of them have a shorter diffusion rate than this one. 2 cells that are smaller that occupy the same physical space are always going to be more efficient and have an increased surface area to volume ratio than the large one. Okay? So as a cell grows, its volume increases faster than its surface area. That leads to a decrease in the overall ratio, as I've demonstrated in previous slides, right? 2 small cells, much like I did here, that occupy the same physical space of a larger cell are going to be more efficient individually, and therefore more efficient as a whole. And because they're more efficient, or smaller cells because they increase cellular efficiency, it leads to the ability to specialize, and cell specialization is vital to the production or for the production of multicellular organisms. Okay, we have around 220 different specific special cell types in our body, and this is only possible because our cells are small. If our cells weren't small, they wouldn't be efficient enough to specialize, and if they couldn't specialize, we couldn't do specific cellular tasks that we kind of utilize our different tissue types for. Like respiratory tissue, lung tissue is really good at diffusing oxygen and CO2. Blood cells are really good at transporting oxygen and carbon dioxide to our cells. Our small intestines are really good at absorbing molecules, right? I mean, all these cells have specific jobs, but these specialized tasks come about as the result of increased efficiency. Increased efficiency comes about as the result of being small and being efficient. Cell compartmentalization. So that was cell division. Cell compartmentalization is all about making special compartments that allow the cell to be more efficient, right? So instead of filling up all of the cytoplasm with all the chemicals and all the molecules that cell needs to live, it makes all of these special compartments with their own membranes. We call them organelles, that all have their own specific job, their own specific function, and because they have a specific function and a decreased volume to do that task within the whole of the cell, it only further increases the efficiency of the cell, because now, instead of utilizing the whole cytoplasm real estate for the creation of ATP, for instance, which is obviously the role of mitochondria, it can just employ the mitochondria in these very small confined spaces to make ATP. Very efficient, very small volume, very large surface area because of the folding that we'll talk about in a little bit, right? But because these cells can compartmentalize, it only adds to the efficiency of the overall cell, which in turn adds to the efficiency of the multicellular organisms. So, organelles have a specific role within the eukaryotic cell. They have obviously a smaller volume than the overall volume of the cell, And because they have a smaller volume, they are more efficient at completing that task than the overall volume of the cell would allow. And therefore, the completion of those metabolic tasks is only increased in terms of efficiency, right? So the fact that cells can divide to become smaller, the fact that cells can compartmentalize into the organelles that make up specific structures that have specific functions, increases to the surface area to volume ratio, and therefore increases the efficiency of the cell. But another way is membrane folding, right? Membrane folding, we see it all over the place. Whether you're talking about the rough ER, whether you're talking about mitochondria, whether you're talking about Golgi apparatus, whether you're talking about the smooth ER, all of those organelles have one thing in common, and they are an interconnected, often interconnected, sometimes not, interconnected membrane, meaning that they are an actual membrane that has been elongated and folded up, and it's been folded with one purpose in mind, and that purpose is to increase surface area. Okay? It is amazing how much actual membrane real estate you can get into a confined space like an organelle by just folding it over and over and over and over and over and over and over, right? So that increases surface area substantially while also decreasing volume. And you know that if you can increase surface area while simultaneously decreasing volume, it only leads to a greater overall ratio. Okay? So, fold your membranes, increase the surface area, decreases volume, substantial increase to the ratio. Really, really important for cellular efficiency. So that was membrane folding. And then the next one, much like membrane folding, is this idea of long, thin structures. And that can be seen very easily in roots, right? When you look at roots, either kind of zoomed out or zoomed in, because all of these individual small roots have even smaller hair projections called root hairs. And those are just to further maximize or increase surface area. So every single one of those is going to increase surface area, and the whole goal obviously in root development and root structure is to maximize surface area so that we can maximize water, we can maximize mineral ions, we can maximize elements, we can maximize nitrates, and we can maximize phosphates, and all of those things that the plant needs to utilize in order to grow. All of those can be more easily diffused in by maximizing the surface area. So by being slender and by being long and skinny, it maximizes surface area. Again, increases surface area while decreasing volume, makes for a substantial increase to the surface area to volume ratio. One thing that we're going to talk about later in the syllabus, but I'm also including it now, is this idea of the intestinal villi and microvilli. Whether you know it or not, when you look at the actual lining of your small intestine, you actually have all of these really, really microscopic, very thin microscopic projections. Not only is your small intestine kind of ribbed like this to maximize surface area, but it also is studded with all of these villi, and you can see them right here, right? What are all of these for? It's to increase the amount of surface area that is exposed to do the function that the small intestine has been employed to do, and that is all about absorption. The higher the surface area of your small intestine, the higher the absorption, and that increases the higher the efficiency, right? You can actually absorb more and more efficiently absorb more by increasing your surface area. So, all of these structures are seen throughout the world of biology, and all of them are really designed to do one thing. They all have one thing in common. That thing is to increase surface area and decrease volume, because that is essential in increasing the efficiency of the cell, of the task, of the function of that cell, which is vital as we go into cell specialization and multicellularity. That's the end of the video. Bring your question to class. See ya.